Namaste students, Khushro Betia, I, JV and Dr. Y. Chandrakala, Assistant Professor, Jyoti Vidyapeeth, Women's University, Jaipur. In today's session, we are going to discuss about the meiosis. As you have aware with the organism reproduce basically two different ways, that is asexual reproduction and sexual reproduction. In asexual reproduction, a single parent usually splits birds or fragment to give rise to a two or more individual. The individual produced by the ways like through the process of mitosis or therefore the same number of chromosomes as that of their parents. So the identical genes of therefore inherit the trait and similar to that of their parent organism. In contrast, the sexual reproduction involves two parents, each of which contribute a specific sex cells. In case of the animal and plants, the male gamete is called the sperm and the female gamete is called the ovum. At the time of fertilization, that the female gamete fuse to form a zygote. Okay, so the zygote divides here through a series of mitotic division to give rise to a new individual. Chromosomes normally exist in pairs, so there are two kinds of body like somatic cells that have a higher plants and animals. 46 chromosomes in human cells constitute 23 different pairs, and the member of the pairing are homologous chromosomes and similar in size, in the shape and position of the centromere. Okay, the diploid number or the 2n. Here the chromosome and the gamete to carry the chromosomes in case like the chromosomal number of the zygote will be 8. That is 4 plus 4 is equal to 8, which is derived from the zygote would contain 8 chromosomes. At the time of the fertilization, when a sperm and egg fused, form and gamete, contribution to a haploid number of chromosomes and a diploid number was restored in a fertilized egg. When the zygote divides by the mitosis to form the first two cells of the embryo, each cell receives a diploid set of the chromosomes that most of the body of the cell is diploid. Okay. So the meiosis consists of a series of nuclear and a cell division. Okay. Each of which so the meiosis contain a series of cell divisions are designated as the first or second meiosis or the simply meiosis 1 or meiosis 2. Each of these includes the prophase, metaphase and the telophase. During the first meiosis division, the member of each of the homologous chromosomes separate and are distributed into a separate cells. In the second meiosis, the chromosome and make up each of the chromosomes separate by the distributed of the daughter cells. The essential process that take place during the meiosis are pairing, synapsis or synaptic of a homologous chromosomes, formation of the chiasmata and the segregation of a homologous chromosomes. From the morphological point of view, the prophase is the first meiotic division is a long process during which the homologous chromosomes pair closely and interchange hereditary material. So the first meiotic phase is divided into the 5 stages, that is liptotene, zygotene, pachytene, diplotene or dikinase. The three stages not sharply demarcated. So here we will see the different stage in details. So the liptotene stage begins when each chromosome is first seen and have a condensed with the interface or the conformation of to form to produce a long thread, or when it is the proteinaceous central axis. Each chromosome is attached at both the end of the nuclear membrane via specialized structure called the attachment plate. The zygotane, the leptotane is considered to be the end of the zygotane stage, begins as soon as the intimate pairing between the two homologous chromosomes is initiated by the process called synapsis or a zygotane pairing. The homologous chromosomes, when paired, are coming from the different sex, like male and the female, and the synapses often starts when the envelope or continue inward in a zipper-like manner from both the ends, aligned to the two homologous chromosomes side by side. The pro-terminal pairing, the two homologous chromosomes starts the pairing at the end or and the terminal, which gradually progresses towards the centromere. The procentric pairing has the centromere and the process towards the end. The intermediate pairing, the pairing may be at the end point of the two homologous chromosomes are drawn together to form a characteristic ladder-like structure called synaptone complex, and that forms a opposite pairing to form a tetrad. As soon as the synaptic complex, along with the chromosomes and the cells, are set to enter into a vegetative stage, the prophase, which are then remain for the day. At this stage, the large recombination nodules, as per the appears at the interval on the synaptone complex. This recombination nodules mediates for the chromosomal change. The non-sister chromatids twist around each other, and the process is called crossing over. So here the beginning of the diploidine stage is marked by the beginning of a separation of the non-sister chromatid and the tight pair of a relaxed. The process is known as desynapsis. The separation of homologous chromosomes is not completed. They remain attached at one or more points where the crossing over has been last for the months or years. So it depends upon the engagement of the RNA synthesis. And in some cases, the chromosome expands enormously, like producing lampresh chromosomes found in the amphibians or some other organisms. So the diploid stage merged into a dikinase. At this stage, the RNA synthesis stops and the chromosome condense, thicken, and become a detached from the nuclear envelope. Each bivalent is clearly seen to condense four separate chromatids, and each pair has a sister chromatid attached to their centromere, where no non-sister chromatid then crossed over, linked by a transmeta. During this phase, the bounding membrane of the centromere degenerates and leaves behind two centroles along with the astrace. Then both of them move apart and take part in the final position or at the opposite poles of the cell. Each centriole is provided with own astral rays. So the nuclear membrane also breaks down and result in the formation of the spindle fiber. So here, during the metaphase, the bivalent chromosome arrange themselves in a plane of the equator, forming an equatorial plane. So the centromere is of each chromosome is directed towards the opposite poles, and the arms of the chromosome is faced towards the equatorial plane. All the bivalent chromosomes repel each other towards the opposite poles, and each pole receives half of the number of the set of the chromosome is brought by the spindle fiber, which similar to the mitotic movement. At this end, the nuclear membranes are found in the endoplasmic reticulum around the group of the chromosome, which appears as the nucleus or a nucleus. The intrameiotic interface. In this stage, the telophase of the first meiotic division and the prophase of the second meiotic division take place. The second meiotic division is essentially similar to that of the mitosis, and it divides in the haploid cells into two dorsal haploid cells, like meiotic division. So here in the prophase 2, does not show the complex nuclear behavior of the prophase and the chromatic and the prophase 2 is widely separated limbs. The metaphase 2 or the chromosome originated on the equatorial plate and have the same relationship to that of the spindle in the mitosis. Anaphase 2, the centromere divide and the two chromatids of the each chromosome separates and move to the poles. After separation, the chromatids are called chromosomes. Telophase 2, at this stage, the reconstruction of the nuclei takes place, as in mitosis. The nucleus of the centriomes and the chromosomes return to the interphase condition. Thank you. This session is powered by 2.0